Great. It's great to be here, and thanks to the organizer for inviting me here. So I'm going to talk about high throughput technologies in cancer research and touch a little bit on synthetic biology at the end as a form of measurement as well. So information flow in the cell. So we were all taught this sort of dogma when we were in high school. And since then, or even then, we understood a lot more about other layers that exist in this flow of information in the cell. There's RNA that regulates other types of RNA as well as itself. There is RNA that we now know regulates DNA and how it functions within this flow of information in the cell. There's, of course, PPI. There is also interaction between proteins and nucleic acids of both types. Also, proteins have modifications that sit on them, such as glycosylation or phosphorylation, that affect the way by which they operate in the cell. DNA also has RNA has changes to RNA as well, and DNA also has modifications such as methylation that determine part of how it operates in the cell. So to understand how cells operate in any context, whether it's disease and health or whether it's any other context, we would like to understand as much as possible of these different layers of this actual molecular flow of information in the cell. So I will try to exemplify that by talking about two cases of jointly analyzing data sets that come from two different levels of this molecular picture of the cell, as well as demonstrate a use of synthetic oligonucleotides in studying aspects of regulation, basically of transcription factors acting on DNA and affecting transcription. Okay, so as I said, I will start with joint analysis. I will talk about using microRNAs and jointly analyzing microRNAs and mRNA, as well as analyzing glycomics data together with expression profiling. But before going there, I will show an old microarray example here that also speaks to the different aspects of regulation in the cell and how the same disease can happen as it can be driven by different changes in the genome, if you will, in this case. So this is comparing expression profiles of BRCA1 mutants to expression profiles of BRCA1 wild-type patients using a very old version of Agilent microarrays. This is from 2002. And as you can see, there's a very nice difference between the expression profile of the mutants and the wild type, but there is one sample here that you want to move to the other side. Okay, so this one is a wild type, but it clearly belongs in the other side in terms of its expression pattern. So of course we observed that at the time as well, and went back and genotyped the patient, and she was indeed BRCA1 wild type. However, she was, I'm not saying that there. Oh yeah, I'm not saying that. So, however, what is happening for this patient is that the promoter of BRCA1 is hypermethylated. So the BRCA1 protein is completely functional if it were to be expressed. But because of the hypermethylation, it is not expressed, and therefore the manifestation in terms of the transcription profile for this patient is the same as a non-functional BRCA1. Okay. And it turns out, as shown in later studies, such as this paper from Chuck Peru, that this phenomenon that we saw as an anecdote, if you will, in this early study, is much more prevalent, and in fact 43% of triple-negative cases have hypermethylated BRCA1 promoters, and this has an association with survival. Okay, so now we're going to move to microRNA. This is a study that we did in collaboration with Annalisa Boris and Dale's group at Radium Hospital in Oslo, covering 100 breast cancer tumor samples. We did matched microRNA and mRNA on Agilent microarrays. And I'm going to talk about the joint analysis of this. Before going to joint analysis, of course, we also looked at differential expression for the microRNAs themselves. Okay, this is microRNA 29, and this is a phenotypic measurement that we do on the tumors, extracellular matrix density. This is extracellular matrix density 1, means that the extracellular matrix is less dense in these patients, and this is 4, where the extracellular matrix is denser. Okay, so we can see that there is, well, I'm not showing that there is because I didn't show you the statistics, but believe me, there is a statistically significant differential expression of microRNAs between ECM4 and ECM1. The top player here is miR29. Okay, so it sort of is differentially expressed between dense extracellular matrix and not so dense extracellular matrix. miR29 was shown to be active in cardiovascular disease. For example, this paper from Eva van Rouge that showed that miR29 regulates, inhibits collagen and therefore leads to less fibrosis, less cardiac fibrosis in mice in this case. And this is a paper from Phil Tsou's lab at Stanford that shows that inhibition of miR29 reduces cases of aortic aneurysm. Aortic is one of the arteries leading to the heart. Aneurysm in mice as well. So miR29 is related to the elasticity of blood vessels, okay, which is very consistent with what we saw about extracellular matrix density. But the fact that it's consistent, that's not enough. We want to know, is this really the case that miR29 is directly affecting the extracellular matrix, or is that an indirect effect of miR29, or maybe some sort of a passenger effect that we're looking at with respect to the ECM? So to answer this question, what we do is we take so this is the 100 patients in the cohort, and I ordered them according to the level of expression of miR29. That's the top panel here. Okay. Now remember, I also have mRNA data in this for the same cohort, for the same people. So now I take all the genes in my mRNA measurement and I rank them according to anti-correlation to this pattern of miR29 expression. Okay, so the top gene here is anti-correlated to this, less anti-correlated, etc., etc. I'm only showing the top, of course, because we have tens of thousands of genes. Okay, so I rank the genes according to anti-correlation, and I'm going to ask, are miR29 targets enriched at the top of this list? So are the targets of miR29 really down-regulated as the level of miR29 expression increases? And therefore, we see a direct action of miR29 on its targets in the tumors. So in order to answer that, are miR29 targets enriched at the top of this list? We're using statistics that we call minimum hypergeometric. I'm going to briefly describe that, even though due to time limitations, I will not go into the details. So the idea is as follows. I ranked my genes according to anti-correlation to miR29. That was the first step. And then some of these genes are targets of miR29, and some are not. The ones are the targets of miR29, and the zeros are not. So I get a binary vector, an ordered binary vector, as a result of this ranking and assigning a 1 or a 0, whether it's a target or not. Okay? Now I look through all possible thresholds in these binary vectors, and at each threshold I count the number of ones that appear above this threshold and compute a hypergeometric p-value for that event. That's easy to do. And then I will take for all these possible thresholds the minimum observed hypergeometric p-value. Of course, doing this trick involves multiple testing, and this number that I get here as this minimum is not really a p-value. Okay, I can correct it, or I can also run a dynamic programming process that actually computes the actual p-value, assuming that I randomly and uniformly draw this vector of zeros and ones. Okay, so again, I don't have time to go into that part, but we can compute an exact p-value for the event of observing this enrichment of ones at the top of the vector. So having done that on this MIR29 data, remember that's where we started, we get the answer yes. Of course, otherwise I wouldn't be showing it here, that the targets of MIR29 are enriched in this set and with the p-value that is significant. Okay, we can also look, in using the same approach, at what functional annotation is enriched in this list of anticorrelatives to MIR29, and we find that in this case it's cell adhesion, again consistent with this ECM observation and with the observations in cardiovascular disease that I mentioned. Another example of that is MIR18. Again, now I will rank according to correlation to MIR18. There's a less obvious mechanistic explanation here, and I'm not going to look for targets when I do the direct correlatives, because of course this is not the mechanism by which microRNA acts, and I find cell cycle to be enriched in the correlatives of MIR18. MIR18 is a known oncomere, so it makes sense that it accelerates the cell cycle. Another example is MIR150. Again, I look at the correlatives, and I find immune response processes to be enriched in the genes correlated to MIR150. MIR150 is orthogonal to the standard molecular classification of breast cancer that was mentioned here this morning. So it is not necessarily highly expressed in luminal A versus basal or anything of that sort. It's completely orthogonal to that, and it is associated to prognosis in triple negative or basal samples. So the next study involves working with glycan profiling. I will explain what that is in a minute. So again, there are two data sets over the same cohort of patients. Glycan profiling is one data set, and the other is the mRNA expression that I mentioned before. We used two cohorts here, again from the Radium Hospital. And let me say what glycan profiling is. So proteins have carbohydrates that are attached to them. They are the glycosylation of the protein. The proteins that go into the serum also have sugars attached to them. What we do in serum glycan profiling is we cleave off the sugars from the proteins in the serum, and we profile only these sugars. Okay, we profile them using an HPLC method, where in this example here I'm showing one chromatogram. So every one of these peaks basically corresponds to a sugar that you need to develop the method in order to associate which sugar it is that's associated to which of the peaks, but that's something that can be done. And in this case, we developed a signature that has, or a pattern, if you will, that in each chromatogram we can identify 46 different peaks pertaining to 46 different sugar groups. Okay, so I have, for each patient, I have the mRNA expression, and I have a vector of 46 numbers, if you will. Of course, the height of the peak represents the abundance of that sugar. Okay. 46 numbers representing the glycan profile, the serum glycan profile. So this is looking in another cohort at cancer samples versus healthy samples. This is glycans in the serum. So this is taken from blood. There is no tumor involved here. Okay? And you can see that there is a reasonably good, it's not as good as we see with gene expression or with microRNAs, but a reasonably good separation of cancer from normal using the glycan, the serum glycans. The top, for example, the top sugar group separating the two classes here is this group 18, and this is the profile for this particular group 18, where the right-hand side are the healthy, and this is the patients with cancer in this case. But Okay, so we can measure serum glycans, and we can maybe say that they could be potentially good biomarkers. But do we understand anything about why it is that we're seeing lower levels of GP18 in the cancer samples than we're seeing in the healthy samples? Can we say something about some mechanism that's happening in the tumor that is reflected in what we're measuring in the blood? So in order to try to answer this question, we can do the same trick as we've done with the microRNAs. And again, here I've ordered the samples according to another sugar group, doesn't matter, GP34 abundance. Okay, I'm going to rank all the genes according to anti-correlation to GP34. And I'm going to do this MAG trick again, finding an enrichment of, so the mRNA, for the mRNA, I do have annotation. I know more about mRNA than I know about these glycans. So I'm going to use that annotation and find which Go terms are enriched in this correlatives to GP34. And in this case, it's cell mobility and cell migration and processes that have to do with how cells, in fact, with how cancer metastasizes. And we know that sugars are involved in these processes of cell mobility. So by using the information that I have about the genes, I can now assign a meaning to levels of certain sugar groups that I see in the blood.
Doesn't that depend on what the person's just eaten and the timing of their own glycogen metabolism?
It does, of course. Well, first of all, this is not glucose in the blood, right? These are cleaved off the proteins.
Okay. But
still, it does. So not all proteins that go into the blood come from the tumor. Some proteins that go into the blood just come from whatever they've eaten. That's true. Nonetheless, if I see a statistically meaningful, so I mean, it's not the case that all the healthy people ate one diet and all the cancer patients ate another diet. So despite this noise, we can still extract the signal. But sure, the serum glycomics is not affected only by the cancer. The point is that the cancer affects sufficiently much of the serum glycomics to be able to get the signal. I should say that I didn't want to go into these details, but the highly sialylated sugars, these are the sugars that have lots of stars on them. This has only one star, but some of them have more copies of sialic acid. They are sort of characteristics of cancer. So cancer normally has more sialylated sugars. So this idea of taking one type of data and jointly analyzing it with another type of data, where I would prefer that other type of data, the mRNA expression for that matter, to have some clear annotation, is something we encapsulated into into a Cytoscape plugin, which is called NVIS. NVIS takes primary data matrix as input, that's the mRNA. It takes an annotation matrix that goes with that primary data matrix, so I know something about each one of these elements. And it takes a pivot data matrix, so there are three matrices that the user has to put as input. And the software, the plugin, computes these associations that I've just shown using that method that I've just shown, and provides all the Cytoscape visualization mechanism to go with it in order to look at the results that you're getting. Okay, and with that, I'm going to shift to synthetic biology. So moving from reading DNA to also writing DNA. So the way by which we make microarrays at Agilent is basically using a device which is like a printer. So it prints, instead of printing RGB colors, it prints A, C, G, and T. And you can think about a microarray as sort of so there are molecules coming out of the floor, and so these are the molecules, right? coming out of the floor. And the first base of all the probes on the microarray is a picture, if you will, in the colors A, C, G, and T. So I will print this picture as the first base. Then, of course, there's chemistry going on, but then I'm coming with the second picture, second base, that's another picture, another picture, another picture, and this way we can grow larger oligonucleotides. We can take these oligonucleotides and cleave them off of the array. And then we get a library of oligonucleotides. This can be tens of thousands of oligonucleotides. And now I'm going to show an example of what we can do with these oligonucleotides. So this is joint work with Ran Segal's lab at the Weizmann Institute. And we used an oligonucleotide library, in this case, to study the sequence determinants of transcription, how changes in sequence in promoter regions can affect transcription in yeast. How did we do that? So, as I said, we synthesized a library of oligonucleotides, whereby there are constant regions to every nucleotide. These are the purple parts here. And then there's the variant region, that's the green. So the green is a promoter region that we have complete control of the sequence of that promoter. Okay, so we can change the sequence content of that promoter to be whatever it is, whatever the questions are that we want to ask about how does this sequence that sits here in the green affects transcription. So, for example, we want to put in the core binding site, we want to not put in the core binding sites, we want to put 2 copies of the core binding sites, we want to maybe reverse the core binding sites. We can do all these tricks. We have, as I said, tens of thousands of questions to ask. And we know exactly what question we're asking because we're designing these variant regions here. So we synthesize these, we cleave them off, we put them into plasmids, which are standard plasmids for that matter, that can go into yeast and express this yellow fluorescence protein from the promoter that we just played with the sequence of. Okay, so now we have, if we had, in this case, we had 6,500 different sequences here. Now we have 6,500 different plasmids. Everyone is driving a YFP from a different sequence that's sitting on the promoter region. Okay, and I want to know which one of them does that better. In which one of them did I manage to not get any transcription at all? Which one of them did I manage to get more transcription than the wild type, for example? Okay, so, and then we take these plasmids, we grow them in E. coli, we put them into yeast. We take these yeast cells and we FACS sort them according to the yellow fluorescence. So I have a bin for every level of yellow fluorescence. There's a red here, ignore it. It's something that has to do with normalization. It's just to confuse you. So I'm going to sort my yeast cells according to their level of yellow fluorescence. Okay? Now I'm going to take each one of these bins and sequence the variants that are in there. So of course I can pull out only the region I'm interested in, and I'm going to ask how many reads do I see from each one of these sequences that I had on my original design. This way I'm going to get for each one of my variants a profile. So K is high yellow fluorescence, 1 is low yellow fluorescence, and I get a profile for every variant. This guy had many reads in the low yellow fluorescence and no reads in the high yellow fluorescence. While this guy had no read in the low yellow fluorescence and many of its reads in the high yellow fluorescence. So I get a profile for each one of the variants. Okay. From that profile, I can now infer a relationship between the variant and the efficiency of transcription, or the efficiency by which that variant drives the YFP, which is what I was interested in. So an example of that would be that we do a scanning mutagenesis of the core promoter of GCN4. So in my variant region here, this is where the YFP is driven, and we have 2 nucleosome repelling sequences here, and in between we have the core binding site for GCN4 in yeast. Okay, this is it. This is the core binding site. And I'm going to mutate this T to all other 3 possibilities, this G to all other 3 possibilities, etc. Then I'm also going to use to play with double mutations and some triple mutations. Of course, even tens of thousands are not sufficient to do a lot of these. Okay, so I have to stop somewhere. And what we see, okay, and this here represents, if you will, the average of that profile that I said I'm getting for every variant. So the more to the right this bar goes, the more efficient that variant is in driving YFP. Now remember, this YFP is driven by GCN4 binding to this, supposedly, assuming that nothing else is happening here. And indeed, this is the consensus, this is the wild type, the consensus sequence, and it has some level of transcription. And nothing else, all the other mutations basically interfere with that transcription except one, this one here. Now, but if you look very carefully at this one here, this is mutating this C into a G. And that's just a reverse complement of the consensus. So in fact, what this means is that it's either the consensus or reverse the consensus, but that's it. Everything else doesn't work. Okay, so GCN4, you don't want to mess with the transcription factor binding site for GCN4. That might not be the case for other transcription factors. Multiplicity, we can put several copies of say, this is again the GCN4, so I have 7 places where I can put it. I can put either 0 copies, I can put 1 copy, 2 copies, etc. For 3 copies, I have 7 choose 3 possibilities of where to put them. I put all of them. And then I study, so every, like, for example, here for, as I said, for 3, I have 35 options. So just the GCN4 is the green part. So I measure all these efficiencies, and I see that indeed, as you would imagine, if I put 2 of them, that's more efficient than just 1 of them. 3 of them is better, but it levels off pretty much when you go to higher numbers. It doesn't help you to crowd more and more core binding sites for GCN4. So we also saw that for GAL4, but that's it. I will just mention that sometimes, you know, if we put them too close to each other, then they interfere with each other. So if you want to drive expression higher in synthetic biology by using a promoter and putting more core binders on it, you may want to be aware of how close they are to each other. Okay. I'll just mention that other groups have used oligonucleotide libraries, such as this paper from Dana Pear and Jay Shenduri, that did scanning mutagenesis of a promoter in E. coli. And I'm not going to go into the results. And we also did work with 3'. So the idea is that using these oligos, you can sort of play with the sequence content of different regulatory elements. I showed promoters and transcription factor binding sites, but it can be anything. It can be regulatory, it can be coding, it can be splicing, it can be any kind of mechanism that you want to study, as long as you have a way by which to measure which of the variants worked better than others, and then that will teach you something about the mechanism that you study. So we did work, as I said, with 3' UTRs. We now moved into mammalian systems, and other labs have also moved into bacterial systems. A recent paper on internal ribosomal entry sites. Protein optimization, of course, you can play with the coding region and see what works better in the protein sense. And we're very interested in new ideas if you have any. So, in summary, I showed some statistics in ranked lists and related tools, including joint analysis of different molecular levels over the same cohort. Glycan serum glycomics is a potential source of biomarkers and a potential source of understanding what's going on in the tumor, which is very accessible to us because it's in the blood. And I showed some applications of oligonucleotide libraries. And thanks to all the people that did the work and to you for your attention.
Well, I have a collaboration proposal. Have you ever considered link RNA binding sites?
No. Yeah, but yeah, do you know the link RNA binding sites?
I think I do.
Okay, good. So yeah, what you need for that is basically some idea of something you want to test sequence determinants of, and some idea of how you want to design the sequences. Because the advantage of this, it's not going to 10 to the 14 different variants. It's under tens of thousands that we know more or less where we want to look. So this is looking under the lamp, but it's a very big lamp. That's the idea. Yeah. So I'm happy to talk about that. Yeah.
Would it work for RNA molecules, single-stranded RNA, or maybe?
We do not synthesize RNA, but the 3' UTR work is in RNA, so we T7 transcribe them, right? Yeah. So you transcribe, we make the DNA. Whoever does the experiment transcribes the RNA, and then yes, you can do RNA.
Thanks, Zohar. And I just have two quick announcements.